Hey team, Dr. Jack Audie here, and today I'm going to be continuing our journey through this AstraZeneca paper. If you haven't watched the previous video, make sure you do, because that gives the breakdown on what the vaccine is, what the study design was, and in this one we're just going to be continuing through the paper. And we're actually only slightly up to halfway through figure 1. So we've got a bit more to go. All right, so the next up in the figure were these heat maps, okay? Let me jump into it. Here are the heat maps here, and so we've got day 0, day 7, day 14, day 28, and we've got some changes in color here. And here we have IgG positive B cells, natural killer cells, and we can kind of see that there's maybe darker shades of red over here. Our CD4 positive T cells, darker shades of red. CD8, maybe there's something going on there, right? And they've attempted to quantify this from their flow cytometry. Over here, we've got a similar expression here, and this is changes from baseline from, again, B cells, CD4 positive T cells, cytotoxic T cells. And over here, this is actually, the cells have been taken out, and they've looked at how much TNF and interferon gamma they produce when stimulated, right? So this is looking at post-stimulation. And again, it's still flow cytometry, but this, they actually stimulated the cells, and that's because cells, when they produce cytokines, they release it, so you don't get to see it in flow cytometry. So you need to take the cells out, you need to block that secretion. I covered this in a previous video. So you block the secretion, and then you can measure whether that cytokine is. Now you can see the big title there, these are bad, and they are. And it's in Nature Medicine, which is a bit disappointed, but I'm happy, and I will put my scientific reputation on the line here to say these are bad, and they are bad for a number of reasons. First of all, the general danger of heat maps, right? So here, this is actual data from an RNA-seq experiment that I've done. But here we have gene A, and here's the expression level of gene A under different treatments, A, B, X, and Y. So A, B, X, Y, and we can see the gene A is expressed at way higher levels than gene B, but there is approximately the same kind of effect going on where Y is way lower than A in the treatments, right? So this is gene B, that's gene A, here are the treatments, A, B, X, Y, A, B, X, Y, and here's the expression level. But you can see in this graph, because it's a good graph, that there is a difference between gene A and gene B, right? They are different because gene A is expressed at way higher levels. It's way turned on compared to gene B. But if we look at a heat map, and this is a real heat map of that data, here's each of these are replicates, so A, B, 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 X, 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 Y, Y, Y. And because heat maps are scaled, and they're often scaled to within row, right? So this is change in expression levels of the gene B relative to gene B. And this is of gene A relative to gene A. And these are Z-scores, which are a statistical approximation. And you can see now that we've scaled it to within gene, we end up with the same heat map, right? So it looks like A and B are doing the exact same thing, but A and B aren't doing the exact same thing, and it might be very important. If this was, say, interferon gamma, and this was IL-4, we would know that there's much more of a Th1 response going on because there's way more interferon gamma than IL-4, right? So absolute differences are important, and we can't just look at relative differences, and heat maps often look at relative differences, right? So heat maps can be very misleading. This is in general. This isn't what this paper did. Just in general, you need to be very careful about when you use heat maps. When we typically use heat maps is when we've got 40 genes, right? And with 40 genes, you don't want to show 40 different graphs. It's overwhelming. You can't interpret 40 different graphs. And if all the genes are kind of related, say they're all 40 inflammatory cytokines or something, you don't need to show 40 different graphs. You can just show 40 different heat maps, and they're very space efficient, right? You can really pack them in, and that's why we use heat maps. But you don't need to do it if you're only looking at a few things. It's much better to show a graph, then we can see the absolute difference. That's a general problem with heat maps. The other problem is scale. Heat maps typically automatically scale to sort of maximums, right? And so there, like, for example, if we think about an infrared camera that uses heat maps to tell temperature, right? So red means hot, blue means cold, right? If we shine it on my face, we can get a blue background and a red face, showing that my face is very hot. But then if we shine it to an oven, we might get the oven is red and the background is blue. Or we could shine it to the sun, and the sun would be red and space would be blue. Because it's scaled, everything comes out as red and blue, right? And so everything can get scaled to the maximum. My face can look red hot, even though it's actually only 10 degrees, 15 degrees warmer than the background, okay? So it really depends on where you set your scales, and your scales will tell the story. So this is the exact same data again. I've just adjusted the scales, and now it looks like there's nothing going on, right? So you've got to be careful with heat maps because they're not telling you an absolute value, they're telling you a relative value, and that relative value is scaled. There will always be a dark red square and there will always be a dark blue square. And so the effect can look very extreme when it isn't that extreme. So that's in general heat maps. This is specifically bad because there was no units. I read the figure legend, I read the paper, I read the methods, and it didn't tell me what one meant, or what 0.1 meant, or what 0.05 meant. These are very different numbers and very different scales, but you can see on each graph there's a very light square and a very dark square, very light square, very dark square, very light square, very dark square. But the difference between this and this is the difference between 1 and 3, and the difference between this and this is the difference between 0 and 0.1. And again, I don't know of what. Like, what are we talking about here? Typically, you would do it as a z-score, and if it was a z-score, you should see somewhere between -2 and +2, but we don't have that. So is this fold control? And if it's fold control, then shouldn't all of these be white because it's fold the 0, and then these should change color? It's all very confusing. So we're missing units, and the units are vastly different between scales, and we don't know what they mean. So that is specifically bad about this paper. Another thing that's very bad is heat maps should never cover summary data, right? And there's a very good reason for this. So here's a heat map. Day 0, day 7, gene A, gene B. So it looks like gene A has gone down a lot because it's gone from here down to here. And gene B has gone up a lot because it's gone from here up to here, right? So we can see the temperature change there. And it looks like a massive effect, right? But this was done on summary data. What do I mean by summary data? I mean you take all your patients and you take the mean, and then you put that mean into your heat map. And the reason why that's bad is we can't see the variation in the data. We don't know how variable the data is, right? You've now summarized it just as a color, and we don't have any error bars to have an idea. So this is that data. And here we can see day 0 to day 7 in gene A, and we can see it has gone down a bit, but when you look at the error bars, that does not look significant at all. And it doesn't look meaningful at all, right? This is probably not significant and not important. And gene B looks even worse. It's the same data. It's gone up a bit here, but remember, it's all scaled. And yeah, so that's definitely not significant. Massively overlapping error bars right there. So you shouldn't do heat maps on summary data. You need to show the individuals. So if we go back to my data, this is my data. I'm showing the individuals. This is an N of 3, A. Each individual here got the treatment A. Each individual here got the treatment B. Each individual here got the treatment X. Each individual here got Y. This isn't summed. I didn't summarize this data. I didn't take the mean of all of these A's, the mean of all of these B's, the mean of all of these X's, and the mean of all of these Y's, and mean them together. So it's very misleading to do a heat map on summary data, which is what they've done. So we don't know the variation in the data. So we don't know how meaningful it is. Is this increase significant or important, or is it barely anything at all? Are those error bars just massive? And I really don't know why they chose to show the data like that. So that's just me ripping into it. I think the story is essentially supposed to be telling you the same thing, that you get an increase in proliferation in B cells because these shades of red are slightly different. You get an increase in proliferation in natural killer cells because these shades are different, and you get an increase in proliferation in CD4 cells right there. But Yeah, it's a bit tough. It's a bit tough. That's what they're trying to say, but that's could certainly work into it. Anyway, now let's go down to the next figure. The next figure, this is when they did the Mesoscale, which is like a cytokine-bead assay, where they measured a whole bunch of cytokines. Now they're comparing two different vaccines here. So they're comparing the SARS-CoV-2 and they're against the meningitis vaccine, right? But it's very important, they didn't take this from the plasma of the patients, right? Here's what they did. You take the blood and you can run it through a density gradient, right? And basically, this medium here, the more dense you make it, as you make it denser, things will float on it. If you think about, the Dead Sea is famously really salty, so we float really well in the Dead Sea. We float much better in the Dead Sea than we do in fresh water we sink, right? So the Dead Sea is a denser liquid because it's full of salt compared to fresh water. So by changing that medium, you change the buoyancy of different things. Now, in here, granulocytes and erythrocytes are very dense. So they can sink in a medium. So we've got this density medium, we've put blood on top, and then we centrifuge it, right? Now, granulocytes, because they contain granules, and these granules are densely packed with proteins like neutrophils full of MPO, and because erythrocytes are packed to the gills with hemoglobin protein, they're very dense. And so when we centrifuge them, they get really heavy, and they sink through this medium, this density gradient medium. The peripheral mononuclear blood cells here, I always call them PBMCs, but these are called PMBCs on this Google image. But anyways, peripheral blood mononuclear cells, these are the lymphocytes and the monocytes. They float. They don't have granules, so they're less dense than granulocytes and erythrocytes, so they float in the suspension. So when we centrifuge them, they come up to the top. So this is what they did these experiments on, PBMCs, peripheral blood mononuclear cells. And what they did was they plated them out in a plate, and then they treated them with spike protein, right? So spike protein from SARS-CoV-2, and then they looked at the cytokine release that came from it. Here's the cytokine concentration on the y-axis, and an important thing to note is this is log scale. So a change in 1 is a change in 10-fold. So even small steps on this graph mean a big difference. I don't know why they chose to do that. I wouldn't have done that because it doesn't clearly show the data. But yeah, so they chose to show it on a log scale. Perhaps it's so they could show IL-8 on the same graph as interferon when IL-8 is expressed massively. And that's very difficult if it's a linear graph rather than a log scale graph. Anyway, so it's a log scale graph. So it's important to know that little steps mean big things. Another thing that's important is that below this line, 10 to the power of 0 is 1. 1 picogram per mil is almost no cytokine at all, right? Cytokines typically work in the tens or hundreds, even thousands, even tens of thousands of picograms per mil. Below 1 picogram per mil, we're not really talking about anything that's worth talking about. That sort of background level, we don't really care about it. So an important thing to note is that below that dotted line is not really worth showing. Interestingly enough, the detection limit of the assay is around about here. So I'm not really sure why they show data below the detection limit on the assay, really. They should just either say not detectable, or, some people put in the minimum values that you can be detected, or 0, and then just not put it on a log scale graph. That's what I would do, and that's what a lot of people do. Anyway, so let's have a look. What are we getting? Now that I've sort of gone into the complications of this graph, what are we getting? So interferon gamma, IL-2, that's a Th1 response. And we can see these stars here mean significantly different. And they'll put a little line there to say that's significantly different to that. Red is the AstraZeneca vaccine. Blue is the meningitis vaccine. Remember, these cells were stimulated with spike protein, so we wouldn't expect really any response in the meningitis vaccine control. And so what do you know? We get a bit of a difference here, which shows that we are inducing a T cell response, and that is much better in the vaccinated compared to the unvaccinated for SARS-CoV-2. So SARS-CoV-2, this vaccine, the AstraZeneca vaccine, has created cells that can recognize the spike protein. So we have had T helper cell proliferation of a T helper cell that has the T cell receptor that can recognize the spike protein. They have proliferated, which is fantastic. And so now they will respond again when we expose them to a spike protein. So we are getting a Th1 response, and so they have caused proliferation in the T helper cell, which is fantastic. There isn't a hint of a T helper 2 response. So they've got IL-4 here, and that's below the 1 picogram per mil limit. So there is no IL-4 being released really at all. So don't even worry about it. There's not a hint of a Th2 response going on here. There is a significant jump in IL-10, so that means there is a bit of a Treg response going on. So we've got a regulatory T cell pumping out IL-10, probably just trying to cool the jets on the Th1 inflammatory antiviral response. And this is very common. As soon as we see a response in the human body, we typically also see the antagonistic response of that. So here we've got an inflammatory Th1 response, and we've got a Treg response, just trying to keep that in homeostasis. Let's not get out of control here, boys. An interesting point here is that these spike proteins have induced a massive amount of IL-8. Now, IL-8 is the neutrophil activating cytokine. It causes degranulation and netosis in neutrophils, and just the spike protein alone in both participants, both vaccinated groups, we've got a load of IL-8. That says to me that IL-8 is acting, that spike protein is acting as a PAMP, a pathogen-associated molecular pattern, and that is activating an inflammatory response within the cells here. Now, that could be a Th17 response. I don't think so, because that requires TCR activation, and we don't have TCR activation in the meningitis vaccinated group. Or it could be from the monocytes, an innate immune response. Those monocytes is a pattern recognition receptor that has recognized the spike protein, released IL-8. This IL-8 is going to cause degranulation and netosis. What is super interesting about that is netosis has been associated with blood clots, because the neutrophils spew out their DNA, and the DNA can go into the blood, where platelets can aggregate into the neutrophils nets, the DNA nets, and that can cause a clot. And now we know that on very rare occasions, the AstraZeneca vaccine can cause blood clots. Now, I'm not saying this is 100% just a hypothesis, because it could be through other means. It could be an autoimmune response going on. There could be a general inflammatory response, which is causing blood thickening. It could be a million things. It could be neutrophils rolling, which can cause clots. It could cause a whole bunch of things. But I'm just saying out there that there is a hint that too much spike protein will cause a strong neutrophilic response, which can be associated with clots. There is a hint of that going on right now. Yeah, so no Th2 response there, and we have that IL-8 response there, which is very, very interesting. It looks like a Th17 response, but it perhaps isn't, because that requires that T cell receptor activation, which we wouldn't get in that meningitis group right there. Right, so that's that. So up next, there is a video to explain a particular kind of ELISA, and that ELISA is an antibody ELISA. And the reason why I'm doing that is I want you to understand that before I go into the next part of the paper, which is all about antibody ELISAs.